Understanding the connection between viral infections and Alzheimer's disease, an in-depth analysis. Introduction. Alzheimer's disease (AD) is a complex neurodegenerative disorder characterized by progressive cognitive decline, memory loss, and the presence of amyloid plaques and tau tangles in the brain. Recent research suggests that viral infections, particularly those caused by members of the human herpesvirus HHV family, may play a significant role in the development and progression of AD. This report explores the emerging evidence linking HHV infections, especially human herpesvirus 6 HHV6, to AD, emphasizing the need for further research into antiviral therapies as potential treatment strategies. The role of viruses in Alzheimer's disease. 1. The infectious hypothesis. For decades, researchers have proposed that pathogens, including bacteria and viruses, could contribute to the onset of AD. This infectious hypothesis posits that such agents may trigger neuroinflammation, disrupt cellular processes, and promote amyloid-beta A-beta accumulation, a hallmark of AD. 2. Herpesviruses of interest. Among various viral pathogens, herpesviruses have garnered attention, particularly HSV-1, Epstein-Barr virus (EBV), and HHV-6. Studies have found increased HHV-6 loads in the brains of AD patients, suggesting a potential correlation between viral presence and disease pathology. Evidence supporting the viral hypothesis. 1. Neuroinflammation and HHV-6. Neuroinflammation is a critical factor in AD pathology. HHV-6 has been shown to activate microglia, the immune cells of the central nervous system, leading to the release of pro-inflammatory cytokines. This inflammatory response can exacerbate neuronal damage and contribute to cognitive decline. 2. Amyloid-beta and viral interactions. Recent studies have suggested that A-beta may have antimicrobial properties, functioning as a defense mechanism against infections. HHV-6 and other herpesviruses may exploit this response using A-beta as a substrate for aggregation, thereby enhancing plaque formation in the brain. 3. Detection of HHV-6 in AD patients. Research has demonstrated the presence of HHV-6 DNA in postmortem brain samples from AD patients. Studies have reported that HHV-6A is more neurotropic than HHV-6B, with increased levels found in individuals with AD. Other studies have linked the presence of HHV-6 in peripheral blood to disease progression. 4. Controversies and contradictory findings. Despite some studies supporting the link between HHV-6 and AD, others have failed to find significant differences in HHV-6 detection between AD patients and control groups. Methodological variations, such as differences in detection techniques and sample sizes, contribute to these discrepancies. Mechanisms by which HHV-6 may influence AD pathogenesis. 1. Autophagy dysregulation. HHV-6 may disrupt the autophagy process, which is crucial for clearing misfolded proteins, including A-beta. Impaired autophagic function can lead to the accumulation of A-beta and tau, furthering neurodegeneration. 2. MicroRNA-miRNA interaction. HHV-6 infection may alter miRNA expression, specifically miR-155, which plays a role in regulating inflammation and neuronal health. Dysregulated miR-155 can contribute to neuroinflammation and neuronal cell death, linking viral infection to AD pathology. 3. Long-term latency and reactivation. HHV-6 typically establishes lifelong latency, which can lead to intermittent reactivation. This process may exacerbate neuroinflammatory responses and contribute to the progression of neurodegenerative diseases over time. Future directions in research and therapy. 1. Antiviral therapies. Given the potential link between HHV-6 and AD, exploring antiviral therapies may offer new avenues for treatment. Preliminary studies suggest that antiviral medications could reduce the risk of dementia in individuals with herpesvirus infections. 2. Understanding viral mechanisms. Further research is needed to elucidate the precise mechanisms by which HHV-6 and other viruses contribute to AD pathogenesis. This includes investigating the role of viral load immune responses and interactions with neuronal processes. 3. Population-based studies. Large-scale epidemiological studies are essential to establish causation and identify specific risk factors associated with viral infections and AD. These studies can help clarify the relationship between viral exposure and disease onset. 4. Biomarkers for early detection. Identifying biomarkers related to viral infections could aid in the early detection of AD and the development of targeted interventions. Conclusion. The association between viral infections, particularly HHV6 and Alzheimer's disease, is an emerging field of research with significant implications for understanding disease mechanisms and developing therapeutic strategies. While evidence supports the notion that viruses may contribute to AD pathology through neuroinflammation and amyloid aggregation, further research is crucial to unravel the complexities of this relationship. As the population ages and the incidence of AD increases, addressing the potential role of viral infections in neurodegenerative diseases could lead to innovative treatments and improved patient outcomes. The topic of the relationship between viral infections, particularly human herpesvirus 6 HHV6 and Alzheimer's disease AD, is rapidly evolving, with emerging evidence suggesting a potential link. This perspective challenges traditional views of AD pathology, which have primarily focused on amyloid-beta, A-beta, and tau proteins. Below are several counterintuitive aspects highlighted in the literature. 1. Amyloid-beta as an antimicrobial agent. The idea that A-beta, traditionally seen as a harmful protein that aggregates to form plaques in AD, may actually serve as an antimicrobial peptide is paradoxical. This suggests that A-beta could have a protective role against infections, including those by viruses like HHV6. This flips the narrative from viewing A-beta solely as a villain in AD to considering it a potential defender against pathogens. 2. HHV6 ubiquity versus specificity. HHV6 is highly prevalent, with estimates suggesting that over 95% of the population is infected. This ubiquity raises questions about its causal relationship with AD, as a virus that exists in most people may not be a specific contributor to the disease. The counterintuitive implication is that despite its widespread presence, HHV6 may play a role in AD only in a subset of individuals who have specific genetic or environmental vulnerabilities. 3. Diverse research findings. Studies show mixed results regarding the association of HHV6 with AD. While some research indicates increased HHV6 levels in AD patients, other studies find no significant differences in HHV6 detection between AD patients and controls. This inconsistency challenges the validity of HHV6 as a definitive risk factor for AD and suggests that methodological differences could significantly influence findings. 4. Potential for antiviral therapies. The increasing interest in antiviral therapies for AD, particularly targeting HHV6, is a departure from conventional treatment paradigms that focus on amyloid-targeting drugs. This shift suggests that addressing viral infections might be a viable strategy for managing AD, although it is still uncertain whether such approaches would be effective universally or only for specific patient subsets. 5. Neuroinflammation and immune response. The role of neuroinflammation in AD is widely accepted, but the exact interaction between viral infection and neuroinflammation remains complex. The counterintuitive notion is that while HHV6 infection may exacerbate neuroinflammation, the immune response to the virus could also play a role in neuroprotection depending on various factors, including the host's genetic background and the specific characteristics of the viral strain. 6. Long latency period. The latency of HHV6 and the potential for it to reactivate decades after initial infection complicates the understanding of its role in AD. This long latency challenges traditional concepts of causation in disease, suggesting that chronic infections may subtly influence neurodegeneration over time rather than being direct triggers. 7. Methodological variability. The wide range of methodologies used in studies investigating HHV6's role in AD, from PCR techniques to serological assays, has led to divergent findings. This variability calls into question the reliability of current data and highlights the need for standardized approaches in future research. 8. Complex interactions with other pathogens. The potential interaction of HHV6 with other pathogens, including other herpesviruses, suggests a multifaceted approach to understanding AD. This complexity implies that a singular focus on HHV6 may overlook the broader context of infectious contributions to AD, emphasizing the need for a holistic view of neurodegenerative disease etiology. In conclusion, while there is increasing evidence linking HHV6 and other viral infections to Alzheimer's disease, the relationship is complex and counterintuitive. Traditional models of AD may need to adapt to incorporate these emerging insights, potentially reshaping treatment paradigms to include antiviral strategies alongside existing approaches targeting amyloid and tau pathology. Further research is crucial to clarify these relationships and to develop effective therapeutic interventions. The emerging evidence suggesting a link between viral infections, particularly human herpesvirus 6 HHV6 and Alzheimer's disease AD, presents a potentially groundbreaking shift in our understanding of the disease. While many studies have explored various factors contributing to AD, the focus on viral hypotheses, particularly the role of HHV6, is a relatively underappreciated area that warrants closer scrutiny due to its significant implications for treatment strategies. Key insights often overlooked. 1. Historical context of infection hypothesis. The notion that infections could contribute to AD dates back to the early 20th century. Alois Alzheimer himself suggested a potential infectious etiology for the disease. This historical context is often overlooked in contemporary discussions, which tend to focus on the amyloid cascade hypothesis. Understanding the historical basis for the infection hypothesis provides valuable insights into the evolving nature of scientific thought regarding AD. 2. Mechanistic insights. Research indicates that HHV6 may influence AD pathology through several mechanisms, including neuroinflammation. HHV6 can activate microglia and astrocytes, leading to neuroinflammation, which is a hallmark of AD. This inflammatory response can exacerbate neuronal damage and promote amyloid-beta A-beta accumulation. Disruption of autophagy. The role of HHV6 in disrupting autophagy, a process crucial for clearing A-beta, is significant. Impaired autophagy can lead to the accumulation of toxic proteins, furthering neurodegeneration. The interplay between viral infection and autophagic processes is a critical connection that requires more attention. miRNA regulation. The dysregulation of microRNAs, particularly miR155, in the context of HHV6 infection may contribute to AD by modulating inflammatory responses and neuronal apoptosis. This connection between viral infection and miRNA expression is a nuanced aspect of the infection hypothesis that could lead to novel therapeutic targets. 3. Contradictory findings. The literature presents mixed findings regarding the role of HHV6 in AD, with some studies showing significant associations while others do not. For instance, while some studies found increased HHV6 loads in AD patients, others, like Alnut et al., reported no significant difference in HHV6 detection between AD patients and controls. This inconsistency highlights the need for standardization in methodologies and further investigation into the conditions under which HHV6 may contribute to AD pathology. 4. Peripheral infection and immune response. HHV6's presence in peripheral blood and its possible reactivation may have implications for neuroinflammation in the central nervous system (CNS). The relationship between peripheral infection, immune response, and CNS pathology is a critical area that remains underexplored. Understanding how peripheral infections could influence brain health is essential for developing comprehensive treatment strategies. 5. Antiviral therapies. The potential for antiviral therapies to alter the course of AD is a significant but under-discussed avenue of research. The current emphasis on targeting amyloid plaques may overlook the value of addressing underlying viral infections. The successful application of antiviral treatments in reducing dementia risk in patients with herpes simplex virus infections suggests that similar approaches could be tested for HHV6 in AD. 6. Epigenetic factors. Recent studies have suggested that HHV6 may integrate into host DNA, potentially influencing gene expression related to neurodegeneration. The implications of viral integration and its epigenetic consequences on AD pathology are complex and often overshadowed by more traditional genetic and environmental risk factors. This line of inquiry could lead to a better understanding of how chronic viral infections contribute to AD. 7. Multifactorial nature of AD. The multifactorial nature of AD is increasingly recognized, yet discussions often focus narrowly on amyloid and tau pathologies. The integration of infectious agents like HHV6 into the broader framework of AD etiology underscores the need for a holistic approach to research and treatment that encompasses genetic, environmental, and infectious factors. Conclusion. The growing body of evidence linking viral infections, particularly HHV6, to Alzheimer's disease is significant and calls for a paradigm shift in how we understand and treat this complex condition. Acknowledging the historical context, exploring mechanistic insights, and emphasizing the potential for antiviral therapies could pave the way for innovative approaches to AD prevention and treatment. Future research should focus on establishing clearer causal relationships, refining detection methods, and developing targeted therapeutic strategies that encompass the multifactorial nature of Alzheimer's disease, including the role of viral infections. 1. Viral infections in Alzheimer's disease AD pathogenesis. A growing body of research suggests a significant link between viral infections, particularly human herpesvirus 6 HHV6 and other herpes viruses, and the pathogenesis of Alzheimer's disease. Studies have shown that HHV6 can induce neuroinflammation, disrupt autophagy, and potentially seed amyloid-beta A-beta aggregation, which is a hallmark of AD pathology. The infectious hypothesis posits that these viruses may play a role in the initiation or progression of AD by influencing neuroinflammatory processes and contributing to the accumulation of amyloid plaques in the brain. This connection raises the possibility that targeting these viral infections could be a novel therapeutic strategy in preventing or treating AD. 2. Controversial research findings and methodological variability. Despite the emerging evidence supporting the link between HHV6 and AD, the research is marked by significant controversy and variability in findings. Some studies provide strong evidence for increased HHV6 presence in the brains of AD patients, while others fail to find a significant difference compared to controls. The discrepancies often stem from differences in study design, sample size, and methodologies used for detecting the virus, e.g. PCR. RNA sequencing. This inconsistency highlights the need for standardization in research approaches and the development of robust criteria for establishing causation versus correlation in the context of viral involvement in AD. 3. Role of microRNAs in viral infection in AD. The dysregulation of microRNAs, miRNAs, particularly miR155, has been implicated in the relationship between HHV6 infection and AD. HHV6 is known to suppress miR155, which plays a crucial role in regulating neuroinflammation and microglial activation, two processes central to AD pathology. Elevated levels of miR155 can lead to increased production of pro-inflammatory cytokines, which exacerbate neurodegeneration. Understanding the interplay between viral infections and miRNA regulation could provide insights into the molecular mechanisms underlying AD and offer potential therapeutic targets. 4. Antiviral therapies as a potential treatment avenue. Given the compelling evidence suggesting a link between viral infections and AD, there is a growing interest in exploring antiviral therapies as a potential treatment strategy. Initial studies have indicated that antiviral medications may reduce the risk of developing dementia in individuals with a history of herpes virus infections. Ongoing clinical trials, such as those investigating the efficacy of valacyclovir in AD patients, aim to determine whether targeting these viral infections can lead to improvements in cognitive outcomes. The success of such trials could pave the way for new preventive and therapeutic approaches in AD management, emphasizing the importance of addressing infectious agents in the context of neurodegenerative diseases. 1. The Spanish flu and its aftermath. The 1918 influenza pandemic, known as the Spanish flu, caused significant mortality and is now being studied for its long-term effects on neurological health. Research suggests that survivors may have an increased risk of neurodegenerative diseases, including Alzheimer's. In the wake of the pandemic, the correlation between infectious diseases and chronic neurological conditions became a point of investigation similar to the current inquiry into herpesviruses and Alzheimer's disease. 2. The link between syphilis and neurosyphilis. In the late 19th and early 20th centuries, syphilis was recognized not only as a sexually transmitted infection but also as a cause of significant neurological decline termed neurosyphilis. This condition illustrates how a viral or bacterial infection can lead to long-term neurodegenerative effects similar to the proposed implications of herpesviruses in Alzheimer's disease. The evolution of treatment and understanding of syphilis' impact on neurological health has parallels with current research into the role of HHV6 and other viruses in Alzheimer's pathology. 3. Mad cow disease and the role of prion. Bovine spongiform encephalopathy (BSE) or mad cow disease led to a public health crisis in the late 20th century. With prion, infectious agents composed of protein, causing neurodegenerative disease in humans. The investigation into how prion lead to neurodegeneration and the resulting cognitive decline echoes present research into the role of human herpesviruses in Alzheimer's disease. Both scenarios highlight the potential for infectious agents to trigger or exacerbate neurodegenerative processes, emphasizing the need for more research into underlying mechanisms. The future outlook for Alzheimer's disease AD research, particularly concerning the role of viral infections such as human herpesvirus 6 HHV6, suggests a transformative shift in understanding and therapeutic approaches. As evidence mounts linking viral infections, especially from the herpesvirus family, to the pathogenesis of AD, several predictions can be made regarding the future landscape of research, treatment, and public health policy. 1. Increased focus on viral etiology in AD research. The hypothesis that viral infections contribute to AD will gain more traction. Researchers will likely prioritize examining the mechanisms by which viruses, particularly HHV6, influence neuroinflammation, amyloid-beta aggregation, and other pathological processes associated with AD. Expect a surge in studies utilizing advanced techniques such as single-cell RNA sequencing, proteomics, and imaging to clarify how these viruses interact with neuronal and immune cells in the brain. 2. Development of antiviral therapies. As the connection between HHV6 and AD becomes clearer, the pharmaceutical industry may invest in developing antiviral treatments specifically targeting herpesviruses. Clinical trials assessing the efficacy of existing antiviral medications such as valacyclovir in slowing cognitive decline in AD patients could emerge. These therapies would aim to address the viral load in the central nervous system (CNS) and mitigate the neuroinflammatory response associated with viral infections. 3. Personalized medicine approaches. Research may lead to personalized treatment strategies based on individual viral profiles and genetic predispositions. For instance, patients with a history of herpesvirus infections or certain genetic markers like the apoepsilon 4 allele might benefit from tailored antiviral treatments combined with conventional AD therapies. This approach could enhance treatment efficacy and improve patient outcomes. 4. Public health initiatives on viral education and prevention. With growing evidence linking viral infections to AD, public health campaigns may focus on educating communities about the potential risks associated with certain viruses. Vaccination or preventive measures against herpesviruses could be advocated, particularly for high-risk populations. This could lead to a broader understanding of the interplay between infectious diseases and neurodegenerative disorders. 5. Interdisciplinary research collaborations. The complexity of AD necessitates interdisciplinary collaborations among virologists, neurologists, immunologists, and epidemiologists. Research consortia may form to explore the multifaceted interactions between viral infections, host genetics, and environmental factors contributing to AD. This collaborative approach will likely foster innovative research designs and comprehensive understanding of disease mechanisms. 6. Novel biomarker development. As the understanding of HHV's excess role in AD deepens, researchers may identify specific biomarkers associated with viral infection and AD progression. These biomarkers could facilitate early diagnosis and help track disease progression, enhancing the ability to intervene before significant cognitive decline occurs. 7. Evolution of AD clinical trials. Clinical trial designs may evolve to incorporate viral status and immune responses as critical endpoints. Trials may assess not only cognitive outcomes but also viral load and immune markers, providing a more nuanced understanding of how antiviral treatments influence AD pathology. 8. Potential for broader implications in neurodegenerative diseases. The findings related to HHV6 and AD may have implications for other neurodegenerative diseases. Research could expand to explore the roles of viral infections in conditions like Parkinson's disease, frontotemporal dementia, and multiple sclerosis, potentially revealing common pathways influenced by viral pathology. Conclusion. The future of Alzheimer's disease research is poised for significant change, particularly as the viral hypothesis gains momentum. With advancements in technology and a growing understanding of the interactions between viruses and neurodegenerative processes, there is potential for novel therapeutic strategies that could alter the course of AD and improve the quality of life for millions affected by this devastating disease. The ongoing dialogue within the scientific community regarding the infectious hypothesis will be crucial in shaping future research directions and therapeutic approaches.